Hi there. Welcome to Cats in the City by Sith Sens. I'm Johnny.
And I'm Pearl.
Together, we would like to share with you bits and pieces of knowledge that we find interesting.
And could be of use to you in the future.
Pearl, can you help me out here, please?
Yes, how may I help you?
I'm looking at this slide, under the microscope, but I don't know what am I looking at.
Okay, let me have a look.
Here you go.
I see. This is a thin blood film, stained with Giemsa stain.
Yes. I've prepared this thin blood film, and then, stained it with Giemsa stain.
I supposed, you are looking for malaria parasites in the blood?
Yes, you're right again. But, I don't know what am I looking at.
It's alright, let me show you. These are normal red blood cells, RBC.
But, there are other things in some of the RBCs. What are they?
Those are RBCs infected by malaria parasites.
That's what they are.
Let's zoom in on one of the infected RBCs. We called them host RBC. And that's the malaria parasite inside the host RBC.
That small thing, that looks like a ring, is a malaria parasite?
Yes. That small thing, as you called it, is a malaria parasite. It's actually called a trophozoite. And it's in a ring form.
I get it now. A ring-form trophozoite.
A ring-form trophozoite is made up of a few structures. Firstly, there's a chromatin dot. And a cytoplasm. And finally, a vacuole.
Yes, I'm beginning to see the picture now.
Sometimes, you will see dots spread across the host RBC.
Like this one.
They are actually the stippling of the cell membrane of the host RBC caused by the infection.
Your notes mention many kinds of stippling.
Yes. There are the Schaffner's dots, Maurer's clefts, James's dots, Sinton and Mulligan's stippling, etc., etc.
So many kinds of stippling. How do we differentiate them?
Well, actually, you don't see all these stippling dots in most slides. So, you don't really have to differentiate them.
Phew, that's a relief.
Now, let's go back to your original slide. Here, you can see two special ring forms, an applique, and a double chromatin dots.
What is an applique?
Applique form, it looks like the trophozoite is stuck to the host RBC cell membrane. It's also called a coal.
And the double chromatin dots, it looks like a headphone.
Both of the forms have thin cytoplasm. These forms are characteristics of Plasmodium falciparum.
Ah, so, this is the infamous Plasmodium falciparum.
You should also notice that the size of the RBCs are similar in both infected and non-infected RBCs.
Are there any other specific characteristics?
Yes. In Plasmodium falciparum infection, you will almost always see RBCs infected by multiple trophozoites. This is one of the reasons why Plasmodium falciparum infection usually causes high parasitemia.
What is this banana-shaped cell in your notes?
This is the Gametocyte. FYI, this is the most specific characteristics of Plasmodium falciparum.
So, if I see a banana-shaped gametocyte, the infection has to be Plasmodium falciparum?
Correct.
I remember from our microbiology class, gametocyte is the sexual reproduction stage.
Yes, you are right. If it was sucked by a feeding mosquito, it will initiate the sexual cycle and produce the next generation of new parasites.
Wow, that's a lot to take in. And it's only one of the malaria parasites infecting human beings.
Yes. Why don't you try to summarize the key points in identifying Plasmodium falciparum?
Okay, I'll give it a try.
Let's start with the size and shape of the host RBC.
The size and shape is normal, similar to uninfected RBCs.
The trophozoites?
The trophozoites have thin cytoplasm, single or double chromatin dots, and we can also see applique or a coal form. RBCs can be infected by more than one trophozoites. As such, the infection usually ends up with high parasitemia. We can also see the gametocytes, which are banana-shaped.
How about the other forms?
There are others?
Of course. Let's look at Plasmodium malariae.
That trophozoite looks like a bird's eye.
Sometimes, you will see this form of trophozoite. But, usually, you will only see the usual, single chromatin dot ring form.
I also know that Plasmodium malariae usually has low parasitemia.
Yes, you also seldom see host RBC infected with multiple trophozoites.
Now, this doesn't look like a ring at all.
This is a late trophozoite, it's in the band form.
And, band form, is specific to Plasmodium malariae?
Yes, and no. Other Plasmodium species also produce something similar to band form, but you almost never see them in a blood film.
Okay, I'll keep that in mind.
This is a schizont, containing 6 to 12 merozoites.
Schizont is the asexual reproduction stage, and merozoites are daughter cells, produced through simple cell division.
When mature RBC schizont burst, it will release merozoites into bloodstream, which will infect other RBCs.
What about the gametocyte?
Well, apart from Plasmodium falciparum, all the others gametocytes look alike. They are compact, and almost filled the host RBC.
Let's move on to the other Plasmodium species.
Here, you can see that the host RBC is markedly larger than the normal RBCs.
It's the same for its gametocyte.
True. The increase in the size of the host cell is a specific characteristic of Plasmodium vivax and ovale infection.
So, how do we set them apart?
Through their shape. In late Trophozoite stage, both the parasite and host cells look amoeboid. Whereas, host cell is oval, and forms fimbriation in Plasmodium ovale. Its Trophozoite stays compact.
The number of merozoites in a schizont is also different. Plasmodium vivax has the second most merozoites after Plasmodium falciparum, at 12 to 24 merozoites.
While Plasmodium ovale has 4 to 16 merozoites.
Let's check my notes, again.
I would also add that Trophozoites of Plasmodium vivax and ovale usually have thick cytoplasm and single chromatin dot.
And, we don't usually see multiple trophozoites per RBC.
And, Plasmodium vivax and ovale usually causes low parasitemia, as well.
That just about wraps it up for today.
Wait. There's more.
What?
There's a fifth parasite. Plasmodium knowlesi. It's a rising threat in Southeast Asia.
OMG. How do I identify it?
Well, you see, the early trophozoites look a lot like Plasmodium falciparum. However, the later stages look more like Plasmodium malariae.
How do I differentiate it from a mixed infection of Plasmodium falciparum and malariae?
An expert malaria microscopist may be able to differentiate them. But it usually requires a PCR test to confirm it.
That's all from us this time. I'm Johnny.
And I'm Pearl.
Sharing with you bits and pieces of interesting knowledge from cats in the city.
Thanks and see you.